So good afternoon all. So today we are going to, what practical we are going to demonstrate today? You have any idea? Huh? Extraction of chitosan protein. Protein we are extracting or not? How will extract protein? Isoelectric precipitation. So if we are using acid, whether we can isolate it, precipitate it or not? Yes? Okay. So there are three different samples are there. You might be familiar with this thing, no? Crab shells, right? This is crab shell, and we didn't took these legs because anyway it's edible. So after the remaining portion we took, and what is this? Huh? Then what is this? Shrimp. Okay. So this is the shells of the prawns. So as you can see, it is quite bigger in size, and shrimp will be smaller. Okay. So as sir already explained, like how we are going to extract it. So anybody know? Like anybody explain in a brief how we are going to do this procedure? First of all, what is the first step? What is the first step? Okay, so we have the waste sample. Okay, so how many among you are vegetarians? Like you are not able to handle these things and all? Pure vegetarian. Okay, okay, fine. So anyway, for the further days we are going to do different practicals. So we have to construct minimum 3 groups. Okay, so for the 3 groups, you guys, like whoever who can't handle these things, they can join in that group, and you can see how the extraction procedure is ongoing. Okay, anyway you will be getting gloves and you will be getting aprons. So who are interested to come, they can come and do together. Anyway you are going to do these practicals, not us. Okay, we will be showing how we can do. Okay, so first of all we have collected this sample from Odisha, like your Rourkela nearby market, and we have dried the sample at 60 degrees Celsius for 3 days. Okay, so the moisture content is reduced as much as possible. Okay, so it is highly dried matter. So why we are going for drying? Because we can store it for a long time. Apart from that, it is very easy for extraction process. Okay, otherwise the moisture content is there. If it is dried matter, it is very easy to extract. Okay, so first of all, what process we are going to do? Anybody? We have different reagents. First of all, we thought of taking acetic acid, citric acid, and HCl. So it will consume a lot of time, and lack of students. We will be taking only HCl and acetic acid. HCl is a hydrochloric acid, which is a organic or inorganic acid. Inorganic acid, and acetic acid is a organic acid. So we will be comparing like the effect of two acids and how the extraction happens. So first of all, we will be treating with, even initially we thought of taking different concentration. So it will be too long procedure. So instead of that, we will be taking 2%, not 2 molar, 2%. 2% whether it's 2 molar or not? No. Okay. So 2% HCl we are going to take. So how we will prepare 2% HCl? 2 ml 98 ml of distilled water. Okay, so we will be having distilled water sample. Okay, so distilled water sample, and we will be preparing anyway. We already measured the HCl, otherwise it will be difficult for you. Usually we will add acid to water or water to acid? Add acid to water. Why? If you are immediately adding water to acid, then it will, fumes will be coming and it will be spilled off to our body. It will be very difficult. That's why we will be using acid water. Okay, we have taken the acid already measured. You can slowly add it to the measured jars. We have 6 beakers we arranged, and 3 different sample. So you can divide it into 3 groups, and each group will be getting one sample. Okay, and you can do it with both acid. Okay, like acetic acid and hydrochloric acid. First of all, we will be adding with this shrimp with or the shell of each of them with each of the acid. So what happens during this treatment? Any idea? Huh? Bubbles? Okay. What process happens chemically? Demineralization. Okay. So what we will obtain after demineralization? Calcium. For example, if we have hydrochloric acid, then calcium chloride. Okay. If we have acetic acid, then calcium acetate. Okay. So demineralization then. So when the supernatant and the retentate will be there. So what we will obtain? The white remaining matter. What will be the remaining matter? Whether the protein will be dissolving or not.
Dissolving.
Protein will be dissolving. Dissolve. Any idea? No. We are not precipitating with isolated pH. So how it will dissolve or not? So okay. So after that, what we are going to do after this process? We will be discarding the HCl or not? Or okay, we will be discarding that HCl. Then the bottom there is a dry matter will be there. So we will be simply crushing. We won't be powdering it. Okay, we will be simply crushing. Anyway, we are dissolving acid, so definitely it will dissolve. Definitely, no need of further crushing. So after that, we will be getting the bottom matter. So we have to wash it multiple times. Okay. So meanwhile, you can once we kept it for almost 2.5 hours. After that, we will be going for the remaining powder for analysis. So meanwhile, we will be kept it for some times. After that, we will be what is the next stage? After that, what we can do? We will be washing with water at least 2-3 times for the removing all the debris, right? So after removing all the debris, what we can do? Drying. We can dry the sample, right? We can dry it for some times. Okay. So then next one, deproteinization. How we can do deproteinization? Adding alkali, NaOH, right? 0.2 molar NaOH. So that we will prepare meanwhile. Okay. So after adding alkali that NaOH, the protein fraction, where we will get the protein fraction? We will be mixing. We have a powder. We have dried it for some times. We will be adding the NaOH. Then we will be getting what? There is a precipitation will be there, and the dissolved matter will be there. Protein will be belongs to which portion? Dissolved matter. Okay. So that we will dry, and this portion also will dry. So where we will get even after that, next stage? Deacetylation. How we will do deacetylation? Okay. How? Acetylation, chemistry, everybody learned. Okay. Okay, remaining process we will discuss in details. Okay. So as of now we will discuss this portion. Sorry, we will do this demonstrate the experiment. So anyway, you are belongs to some people belongs to particular institute. So try to mix up together to interact among yourself. Make a group. Okay. So we need 3 groups, and whoever interested, we will be giving the apron and gloves and the hair cap as well as the glass. Okay. For one among you, whoever interested. If 2 people can do, 2 of you can also do that. Okay. So just come forward, whoever interested, we will be giving. So you can do these things. First of all, crush it among yourself. Then we will be giving the sample and crush it. Then do the experiments. Okay. 950 ml in both of the beakers. Okay. Sorry, 980 ml. Let's open now. Carefully. Close it. Because we are not going to do like critical analysis or estimation for just for digestion. So if 1 ml or 2 ml difference doesn't create any big issue. This much. 480. Okay, that also. So first of all, we will be preparing acetic acid. Okay. So acetic acid, 2 samples will be there. Third one we will prepare later. Okay, it's no matter. So Okay, so these two are 20 ml each. Okay, so here we have 30 ml. So instead of that, we will be taking 20 ml. Okay, approximately. Okay, so we can pour it into bottle slowly because it's as pour it completely. No worries. Okay, so the first acetic acid is prepared. Next one, you can take the 20 ml from this. Enough. That bigger. So you can label it because otherwise we will be confused. So you can label both of them as acetic acid. So this is the 2% solution of acetic acid. Okay, here also. Okay, so the next one we have to prepare HCl. Okay, similarly we will be keeping it aside, and correspondingly we will be making 2 HCl. So the same, so we will be having 114 ml of what? 35% acetic acid, sorry, HCl. So next we have to take the half of them to both of. So what is the half of 114? 72 and 72. So how much ml required to make it into 1 liter? 928. 928. Okay, so take 928 and 928 from each. First 500, then 428. So someone else can do, na?
Right?
Mm-hmm.
114 by 2. 57. It will be 57.
Mm-hmm. Okay, so 57, what is the remaining? 40.
943.
943. So 43.
943. 57.
Next 443.
443.
Take 443 ml.
One.
Okay. 440. Okay. 447, no?
440.
So nearly is approximately fine only.
443.
Fine only. This is 10 different now. This is 440. Okay, fine only. You can pour. It's HCl, so be careful while doing it. Okay. Yes. Otherwise. Okay. So take 57 ml. Take carefully. Okay.
57.
50 will be there. Then take again 7. Nothing will happen. Slowly. And then pour slowly, okay, this spark will come. Pour the side, like that. Okay. Similarly, fill this bottle as well. Anybody else can do that? Oh, okay. Okay. Let's take 500. Okay, enough. Enough. So you can pour the remaining, it's completely 57, so you can pour the remaining to the side of the beaker, so nothing will happen. Slowly, okay. Careful. Okay, fine. Okay. So there are two different samples, acids we have prepared. The next one we will prepare for the third one. So anyway, so you can take the sample like whatever waste of the shrimp and those things, we will just crush it and we will be adding in. So first of all, so we have to mark it, which sample it is. Okay, so it is HCl, right? Okay, this also. Okay, so we have to mark which sample we are going to add it, because after dissolving sometime we may not be able to identify. Okay, so the 2% acetic acid, 4 different samples we have, and we will be taking the shrimp. Okay, so whoever handling the shrimp and whoever handling, just use the gloves because they having spines, it may causes toxicity like tetanus toxide may be there, or any other pathogens may get into our body. So this is very difficult. Don't take risk while crushing. So take the sample. So sample. So weighing balance. You can just do yourself.
It's 2% is in it.
Come. So you can wait someone. So how much percentage we are going to make?
1:20.
1 is to 1 is water ratio 1 is to 20 ratio. Okay. So if we have 1 liter, so what is the amount of
50 grams.
50 gram? Okay. So we have to take 50 gram of each samples. So we can wait. We can weigh the samples. Okay. You can see it is purely dried and some other debris, waste matter also there. Okay, so approximately I think 3 samples will be sufficient to make 50. So as you know, it's very sharp edges it will be having, and there is some other matters also there inside. So definitely it may be containing other protein fraction and other fraction. So crab usually contains some amount of sugar. So those crabs are known as sugar crabs. Okay. So these all are from sea water, not from so we will be breaking it just for definitely it won't smells good. Okay. So now someone can wait. Okay. So really can weigh the sample. Okay. We'll go inside, right? No, it's really going inside. Okay. 50 gram. Take more than 50, so 60, 60 into.
लेकिन आई थिंक ज़्यादा हो जाएगा इसमें बताने का नहीं।
Okay. Okay, you can transfer it to here. 10, 10, 10 each. 10, 10 each. First,
Okay,
take
okay.
10. Sir.
और पहले से करना पड़ेगा।
No, fine, fine. Take 10. It's initially tired, right?
What?
Tire is done na, initially.
Yeah, yeah.
Fine, fine. No, that's fine. You don't exactly. Okay. So 10 gram like that way. So first of all, this crab into will be transferred into acetic acid and HCl. Okay. So we can measure 50 gram at a single stretch. So we will be measuring 10, 10 each, and we will be transferring into this. Okay. So transfer it to the beaker. Click. Shrimp sample. Actually, we have less quantity of prawns sample, so that we will do later. So whatever is available accordingly. Crab remaining. Crabs. Carefully. How much?
It's 30.
So total 30 become. Next. Some weight, high weight particle. Put now. So like you might be familiarized with the chitins and chitosan, right? So what is chitin? What's the monomer of chitin? Any idea? So everybody is using chitin, chitosan in packaging materials. Most of their research is like edible packaging. Chitin combined with some nanoparticles and their effect. There are a lot of people who is doing research in that. So in India, like we can, but there are some demerits are there. For example, if you say vegetarians, okay. So for vegetarians, we can't provide the chitins along with their. If I am coating with chitin, what will happen? Some unknowingly non-vegetarians may consume, but it's from an animal source, right? Similarly, gelatin everybody is like, I won't eat egg cake, but even sometimes we will be consuming the products which is containing gelatin also made from animal source, like, so if you are from, even vegan gelatins are there, like carrageenans and all, so it is extracted from algae, okay, brown algae and all. Similarly, this chitin is also prepared from animal source. When most of the arthropods, you might be familiar with arthropods, which have in the exoskeleton. So not only this 50 gram, okay. So we'll be which having the exoskeleton which contain the chitins. Okay, so we'll be transferring the chitin into this acetic acid. Okay. We will be transferring it. So as much as possible we have crushed it, okay. So let's see what's going to happen. Similarly, someone else can measure. This also there. This is also along with this.
Yeah, yeah.
Okay. So total 50, 1:20% solution we have prepared. Total 50 gram in 1000 ml. Okay, which approximately, if we are checking specific gravity, 1000 ml approximately equal to 1000 liter. Okay. So the next one we have to prepare for the same crab we are taking for, but HCl. Okay. If someone else, if anybody is interested, you can measure, because we give you more quantity. Thanks. Gloves. And if we have carbon dioxide, so can you see any changes happening here? Any changes? See, listen. What is happening? Bubbles formation. Which bubbles it is? Like oxygen or carbon dioxide? Carbon dioxide. So just imagine when these materials which react with acids, which produces carbon dioxide, and calcium carbonate also formed. Okay. Then when there is a reaction calcium carbonate with HCl, so what will be formed? Calcium chloride and carbon dioxide will be released. Okay. So similarly,
Can't I have to keep this entire?
fine. That will be better. Okay. You want to use? Oh, this okay.
So 50 gram crab, acetic acid and HCl.
HCl. See, like even apart from that, in general discussion, for example, if you want to use these proteins from these sources, okay, food waste and all. Usually, as we know in India, like most of the industries, most of the dairy processing industry, they are simply discarding the whey. They are not utilizing. Some cases they will be making some whey drinks, and most of the people used to prefer it. So only in India, one and only company who is making whey drinks, whey protein isolate is nothing but Amul. No one is utilizing. Similarly, in the fish processing waste, there are a lot of protein sources are discarding. Okay. For example, most of the researchers they are doing on peptides. Okay, you might be knowing with like a lot of peptides, bioactive peptides are there. From animal source or plant sources, but animal sources is quite stable as compared to plant sources because it's highly prone to degradation, peptides or peptide molecules. So if you want to do any research, for example, bioactive peptides from different sources, so we can easily identify the peptides, and isolation of the peptide is quite challenging. You might need some chromatographic separation is needed. Industrially, there will be like large ion exchange columns are there, but in a small scale it's very difficult to extract peptides, and for like large purpose it is very difficult to isolate the peptides, and industrially we can't utilize in different aspects. So instead of extracting the particular peptides, we can use the entire compounds. For example, a particular biomass is there, and the biomass does not create any other problems, but it's containing some 10 to 20% of peptides, then we can extract the peptides and incorporate it with different packaging materials. For example, different bioactive packagings are there. For example, you might be knowing with the casting method, like for example, how is the casting can be done? If I have citric acid and one starch ester, we can go for casting. If you are mixing the starch with citric acid, that will create some cross-linking. If I am using and if I can dry it, hydrogel, like as you know, hydrogel, cross-linking of the hydrogel, which create the what? Casting film, okay? And the polylactic acid film, you might be familiarized with polymerization of lactic acid when created into polylactic acid. But even recent some studies are saying like polylactic acid, if it is from a biological sources, even though that also taking long time for degradation, so we cannot consider this as a biodegradable packaging. Even though in these packaging materials we can incorporate, or we can either, you might be knowing with electron spinning method, right? Electron spinning, coating. Okay, so you can coat it with some fibrous coating can be done. For example, industrially some meat are available. Okay, you can coat this meat with some bioactive peptide which is having antimicrobial property, and if you are making some silver or gold nanoparticles and which is attached to the even biosynthesis of whenever we have a bioactive peptides and some nanoparticles, we can make it into nanocomposite, right? Whenever this nanoparticle, silver or gold nanoparticle is there, we will be incubating at a certain particular temperature, we will be getting the bio nanocomposite, and we can incorporate with the packaging material, and that will create some, that will having both, even as we know this silver which inhibit the mitochondria, and definitely that will inhibit the growth of microorganism, along with the antimicrobial peptides also will act on it. Like that way it will work. Similarly, different bioactive peptides from different sources, like even meat sources. For example, every meat, every shelled products which is having highly antimicrobial coating over the shell, especially even tortoise also having the antimicrobial properties, because their skin, internal skins are highly soft, like soft in nature. So in order to protect from external organism, they have lot of bioactive peptide which is coated over the surface of the shell. So if we are extracting the bioactive peptides and incorporating into any meat products, packaging, or like electron spinning or any other electron spraying can be also done to coat over the surface, it can be a good research for each of you. Like you can also work on that. Okay. So the next one, we will be transferring 50 gram. Okay, you can transfer it into. So you can see the difference. This is an organic acid. This is inorganic acid, right? Like HCl. So if you are transferring to HCl, you can see the difference, how fastly it is reacting. So if we have a carbon dioxide sensor, we can identify how much amount of calciums are present in it, calcium carbonates are present in it. Like correspondingly, if you are quantifying the molar concentration of the carbon dioxide which is released, sir already discussed, like biomass, how much biomass is produced which is equivalent to the CO2 which is utilized. Similarly, how much CO2 which is releasing which is equivalent to the amount of calcium carbonate which is present. Similar, we can easily identify. So the change, you can see how fastly it is reacting. Similarly, you might be knowing with the Taj Mahal which is currently suffering from this pollution which is due to the calcium, degradation of calcium carbonate due to the acid rain. So if this small 2% concentration itself creating this much problem, then what happened to the shell fishes which is present in the sea? For example, even as compared to the rain in the sea is more intense than rain in the land. So if we are polluting the sea, polluting the air like this, so it may leads to high amount of precipitation of carbon dioxide which is highly affecting the surface organism rather than internal organism. Especially the formation, like some algae are highly resistant to this kind of acidic change, but other shelled fish are, their shell become degraded and it will lead to the death soon. Okay. So see the difference. In the short, even we kept this about some 10 minutes before, and we kept the HCl recently. So this much amount of carbon dioxide which is released. So that's the difference between organic and inorganic acid, their reactivity. Even though you can see how fastly it is degrading, and after some hours we can see the difference. 2-3 hours we can see the difference how it is. Okay. So shall we move to the next powder analyzer is the
Powder analyzer.
so we'll go to it. Okay. After that we'll come back. Meanwhile we'll be doing after doing that. Okay.
Systems can be, for instance, proteins. They can be, for instance, carbohydrates mixed up together, starch or like that. So now if we talk about foods per se, or if we talk about food systems, they can be either solids in nature, they can be either liquids in nature. Gases, we don't take gases as foods, right? So, but in case of solids, there are certain foods which are, if we categorize solids per se, they can be of two different types of stuffs. One, say for instance, apple. So it doesn't flow. Apple doesn't have any flow properties. So it does not flow from one place to another place, and we don't need to make it flow from one place to another place. But if you have chocolate, for instance, so, okay, chocolate is a solid substance, but when it is melted, it behaves something in between liquids and solids. Okay. And then we have another form of substances, say for example powders, and we take a lot of powders, whey proteins for instance, starch for example, you take Complan for example, you take powdered substances for food stuff and like that. So all these things, when we have to study their properties, one of the properties is for instance flow behaviors. Okay, so you must have read about, for instance, one of the easiest ways to measure the flow properties is by measuring viscosity. It's also a flow property. Okay, so then we move on to, say for instance, smaller order to higher order, which means along the length scales or along the if some experiment is easier, then we move on to tougher experiments and more informative stuff. Like the easiest practicals that we do is, for example, moisture content. Okay, so it's a very easy experiment which gives you, for instance, one type of data. But when we move on along that, moisture, protein, fat, ash, polyphenols, other things, fatty acids, composition, it becomes a little bit difficult. You understand my point? So same way, if we start analyzing the properties of liquids or solids, so we move from, for instance, smaller scales to little bit higher orders where we get more information. So if we talk specifically about liquids, we can analyze liquids by understanding their viscous behaviors. Viscosity means how much a liquid flows, and when it flows, how much resistance is there. So for instance, water, if you put a bottle of water here, it will spread easily. But if you put a bottle of honey and spread it, it takes time, because the internal resistance to flow in honey is more, internal resistance to flow in water is less. Okay, so that is one when we talk about simple fluids. But then there is the mixture of fluids. Say for instance, you have starch mixed with water, you have proteins mixed with water, or you have semi-solid substances. We study their viscous behaviors, or we study their flow behaviors in terms of rheology. Okay? So how many of you have how many of you have just maybe heard about rheology? If you have not done experiments, how many of you have heard about rheology? Have you heard the term rheology? Yes. So there's a difference between viscosity and rheology. Rheology can be, viscosity can be part of rheology. Rheology is the basic or a bigger picture, bigger science, which deals with the study of the flow behavior of different kinds of materials. In viscosity, it's just one parameter, the resistance to the flow of different particular liquids. But in rheology, we study number of things. We can study rheology of liquids also. We can study rheology of mixed matters also. Mixed matters means solids plus liquids. But we cannot study the rheology of solids as such. It's very, very difficult to study the rheology of solids. अगर आपके पास solid protein है, solid अगर आपके पास starch है, solid आपके पास flour है, तो you want to study its rheology, it's very, very difficult. Rheology में क्या करते हैं? Rheology, we have an instrument called as rheometer. Probably you can see that also. We have a rheometer, and we have an advanced rheometer, very, very advanced rheometer, which has an attachment called tribology. Tribology is very advanced study, very, very advanced sciences. I studied tribology when I did my third postdoctorate in UK. So it's a very advanced science in flow behaviors. So my point is, so you can study that flow behaviors of certain types of mixed solids, mixed liquids by using rheology. Okay? So rheology में then there are different parameters that we study. So whether a substance is viscous in nature, whether a substance is elastic in nature, whether it's a composed of viscous and elastic behaviors, Okay, so if we are changing the consistency, what happens to the viscous behavior? If we change the consistency, what happens to the elastic behaviors? If we are increasing the temperatures, what happens to the elastic behavior? So maybe PhD students might know this more. So then, agar hum rheology, how many of you know Hindi? Okay, so if I sometimes speak in Hindi, just let me know. If you don't understand, I can repeat also. So when we talk about rheology, so when we do experiments on rheology, so for instance, my student say he is doing rheology of starch, for example, so he will do rheology of starch in terms of change in the frequency. So we call that frequency dependent tests. And then we have temperature dependent tests, frequency sweep tests or temperature sweep tests. And this rheology is only and only done when we have food which is a paste. Paste means something which has a lot of water in it. Okay? So it could be, for example, fruit purees also. Okay, fruit juices we largely avoid by doing, but there are specialized equipments to do that. Now, what about powders? So we came from liquids to semi-solids or mixtures to now powders. Okay, so how do we study the characteristics of powders? So powders also have to flow from one place to another place. Say for instance, somebody is having a flour making industry or even a starch making industry, and you are filling it into powders. You see these baby foods are coming up in the market. So they will also come from somewhere. They have to be transported from somewhere and then filled into cans. Aisa to nahi hoga. I mean, it will not happen this way that there will be if you ever will get to visit Nestle, a baby food division, it's highly automatized zone because baby foods are sensitive in nature. We cannot, there is no room for the growth of microorganisms in baby foods because they are extremely sensitive foods. So there is no manual intervention in that. It is always done automatically. So what I meant to say is that when those baby foods, for example, LAN 1, lactogen 1, lactogen 2, you might have seen that. Hai? Or if you see, for instance, our skimmed milk powder, which is powder in nature. So it has to flow from somewhere and then get packed in different kinds of stuff, maybe small jars, maybe small cans, or maybe in bigger what we call them gunny bags or like that. Okay, so this powder has to also flow. So you have to understand when we just have a larger picture in mind related to industries, we have to understand the flow behavior of powders also. Flow behavior of maybe in your cereal sciences course or maybe in your this grain technology courses, you might have read about the stacking of grains. So when we stack grains, rice, when we have to stack them for a pretty long period of time, it has a characteristic heaps are formed. Okay, so it also has a flow. It can flow downwards. Okay, that's normal against the gravity. But so we largely study in older terminologies. We used to study the flow behavior of powders through orifices. We used to study the flow behavior of powders through chutes. Chutes means openings. Okay, orifices also means openings. Chutes are openings which are if you have ever seen a grain milling machine. So when we are putting rice in it, it is paddy in it. It is getting milled and rice is coming out. The open sheet like structure is that's referred as chute. If it's closed, it's referred as an orifice. So the powders have to flow through those orifices, or powders have to come out of these chutes. Also, one of the disadvantages with powders is we have liquids, we have semi-solids, mixtures, and we have powders. Less dependency on water, external water, very less dependency on waters, very high dependency on external moisture contents. Agar aapke paas you have got honey, you have got water, it will have lesser impact. If you are storing it for a pretty longer period of time, it will have a lesser impact of external moisture content. Okay, so viscosity, you can do it at any temperatures, any moisture contents. Okay? Now we move on to starch, for example. It also doesn't get so much impacted by external moisture contents. But in case of powders, if external moisture contents, external relative humidity or external humidity varies, your powder flow will vary. You have something at your home, coffee for instance, and in extreme monsoon season, it becomes lumpy, so it cannot flow. Understand? So same way, when you are doing things in industries, you cannot have, say for instance, lumpy solids being pushed through materials. It is not energy intensive process. It's an energy extensive process. So that means you will need more energy, you will need more money in industries. Industries revolve around money, quality. So if something goes wrong, so that means you are investing more money into it. So what I wanted to emphasize here is that the traditional methods for analysis of these powders, for example, through the flow behaviors, through orifices, through chutes, which is very, very well described. And if you want to learn more about it, Singh and Saha, you can refer to it, flow behavior of powders and through different orifices and chutes. It will take almost one hour if I describe all the properties. Okay. So now we have a very, very advanced equipment. Similarly, the way we wanted to analyze the viscosity of liquids, there is an instrument referred as Ostwald's viscometer. Very, very good instrument to measure viscosity of liquids. Similarly, if we would want to measure the viscosity of mixtures, we want to measure the viscosity of pastes, we can measure the viscosity profiles, we can measure the rheology of it by using a rheometer. Now, if we have to measure the profiles, different profiles, there are different profiles. The way in rheology we have temperature sweep tests, we have stress-strain relationships, we have frequency sweep tests. Similarly, in powder flow analysis, we have got number of tests which you will see experimentally when the experiment runs up. So it takes a lot of time to run up the experiment. So Jeshu is a very cool PhD guy we have. So he has handled the equipment number of times. So he will tell you that this is the way we operate it and this is the data that we get out of it. Okay. So but if we run it at that time, it will take almost 60 to 70 minutes. So he has already ran few tests, and while he will just show you that we put samples here, and then we run this way, and this is the data that we get. Okay. So half of the part will be skipped, only that because it takes 60 to 70 minutes. Okay, my point. So now in this powder flow analysis, and the conventional ones. Conventional means traditional processes. Traditional processes of measuring the powder flow behaviors are very tedious, and they are dependent upon human beings. Okay, largely because external factors can come into play. Say for instance, water, moisture content, relative humidity, everything can come into play. But when we do it in these equipments, there is less intervention of human beings, and you can get number of properties in that. Okay, so this is kind of the basic funda behind this powder flow analyzer. The powder flow analyzer that we have is, I think it's from Antonpar, right? Oh, Brookfield. Brookfield also makes viscometers. It also measures very specialized rheometers, and Brookfield is a company based in UK. It also has made this one, powder flow analyzer. So a lot of our students who are working on, say for example, flours from millet or starch from millet or proteins from millet study the flow behavior properties of this powders also by using this Brookfield's powder test analyzer. So you will fill in a lot of these samples, 2 or 3 samples are there, you can just check it out, and it will show you different properties related to the flow behaviors of the powders. Okay, do you have any questions? Okay, are you sure? 100% sure? So if you have to read along this track, along this trajectory, you have to understand first the viscosity, which is very, very easier to understand, and then you have to understand the rheology. Rheology, you can go to Antonpar websites. They have a lot of data on rheology, and also one of the finest guys, his name is Jasim, and one more guy, he is in Sweden. He's also an Indian, Santanu Basu. Two best people in the world who have done extensive work related to rheology. So there's a book on rheology also, which is written by both these, Jasim Ahmed and Santanu Basu. You can refer to those books. And then powder flow, it is mixed in between all these two things. You can read it from anywhere. Okay? So you can go inside and maybe just go like 4 people and just to see the equipment and accordingly to it. If you have any questions anytime after this experiment also, so my office is just in the same block where you have your lectures. You can come to my office anytime. Okay? Perfect. Cheers.
Okay, so this is today we will be doing the powder flow analysis or the powder flow test. So for that, the machine that we will be using is this one, the Brookfield's powder flow tester. And the other things which we will be needing is of course the sample, a powdered sample. It has to be sieved properly, and then we will be analyzing it for it. And then we need this sample trough. This is called the sample trough. Okay, this is where we keep the sample. This is called the inner catch tray. Okay, and there are other equipments like this is called the outer catch tray. In subsequent part, we will be coming to know how that is used, and it comes with 2 lids. First, this is called compressibility lid or the friction lid. Okay, this is perfectly plain as you all can see, and this is called the vane lid. It comes with 18 compartments. Okay, and this is called the profiling blade. So this profiling blade has 2 parts, one curved part and one flat part. The flat part we use with this compressibility vane, compressibility lid, and this curved part we use this with this vane lid. Okay, and there is of course this brush, cleaning brush, spatula to put the samples. Okay, and to run this we need a software called powder flow tester software. We need a system, and in this system already the software has been installed. The software is Powder Flow Pro V1.3. Okay, this software we will be needing. Okay, so now first thing, these are the tests. So this is the first interface that comes up when we open it. Okay, and these are the tests that we can do. Okay, first comes the standard flow test, then comes quick flow test 5 point, quick flow test 2 point. These are to analyze. I will be telling you subsequently why these tests are. Okay, just let me just go through the tests that we will be doing. Okay, then comes standard time consolidated flow test, quick time consolidated flow test, standard valve friction test, long valve friction test, quick valve friction test, and standard bulk density test. So here now we will be doing basically 2 tests, the standard flow test and standard valve friction test. Okay, so for the standard flow test, since we can see the trough and lid, they have mentioned here already the trough and it's fancy. It's better if this. Okay, they have mentioned this trough and lid. We can see in these two are the lid part. Okay, so in this lid part, this has been mentioned. PFT 500. So when we select it, the lid comes towards it by default. It selects, since we are going to do standard flow test. So they have mentioned that we have to use PFT 500. It has 2 engagement tips, and this is the compressibility. This is the compressible lid. Okay, so this will be coming up and down. This will be going up and down. So here we have to fix this. There will be 2 knots in it. This has been perfectly fixed. To remove, here is a hook present here, so we will have to press it and then we have to release it. Okay. Now next thing which comes is we have to weigh this sample trough. First we will weigh this. Initial weight is coming around 720 grams. Okay, 725 grams. It's around like that. Keep this here. First we will have to place the inner catch tray here inside and the outer catch tray. So this is a non-destructive process. That means the sample, once we analyze it using this powder flow tester, we can reuse it. So that's why we are very conscious about the samples quantity. That's why we have this inner catch tray and the outer catch tray. If something falls in the inner catch tray or the outer catch tray, we can reuse it again. Okay. And then now using the spatula. So now since we are going to do the standard flow test, so we will be using the curved portion. Okay. So the flow test is basically for if we look at the silo, we have seen the shape of it. The valves, these are not for the valves. The free flow that occurs in the center. At the center, when the free flow of the powder that occurs at the center, that we will be analyzing using this standard flow test. And for the valve test, we will be using this part. That means the frictions or the stresses that occurs in the valves of the silos. That we will be analyzing for that. So now I will place it here. Okay, so now as you all can see, I have just haphazardly placed the sample somehow. Okay, now we will be making it uniform. We will be making a uniform heap. For that, we just need to hold this profiling blade nicely and just either rotate it clockwise or anti-clockwise. Anyway. Once this is done, as you all can see, a nice heap has been formed. So I will just first I will remove the inner catch tray. Then I will slightly remove the outer catch tray, and we will weigh this sample. Okay, wait, better I tear it once. It's not been teared. So initially, what is the weight that we got of this? 725. Okay, now we will measure it. So it's coming around 845. So it's around 120 grams, right? So 120 grams of this. Now we will place this sample tray down at the bottom. We can fix this here, we will fix it, and then we will have to look for a water bubble. There is a water bubble in it, okay? We have to see whether it is at the center or not, so that the weight is balanced, okay? Now here, the first thing that we will see is the setup. In setup, we will see if this system is connected, it will show a green light like this, and it's saying that it has been connected to port 4. I've already connected it. We can just search and connect it. That's done. Now coming to the test, now we will write the product name. First, we'll name it as GIAN sample 1. I think the caps lock is on. Sample 1A. Okay, done. Now we will write the weight, sample weight. What was the sample weight? 120 grams. Okay, 120 grams. All right. Then we will be going to the analysis part. In analysis part, we will select this raw data, stress data, frictional angle, density curve, hopper design. And after the test, we will be getting the values of consolidation endpoint, major principal consolidation stress, unconfined failure stress, effective angle of internal friction, effective angle of wall friction, bulk density, and hopper half angle. After that, we will be going to comparison. In comparison also, we will select the data of flow function, friction angle, density curve, hopper design. In statistics, we will select the slope, intercept, arcing dimension, critical stress, critical internal angle, critical density, red hole diameter, critical stress, critical internal angle, critical density, fill bulk density, maximum bulk density, compression index, compressibility ratio. Setup has been done, and then we will go there and run the test. Actually, once we'll select run the test, this plate will come down and the process will be done. But that itself will take around 20 to 25 minutes. Normally it takes around 25 minutes. So that will take a lot of time, right? So I will not be showing the running time. Okay. Next, I will be showing you the other thing, which is now once the test has been done. Okay, suppose we don't have to do anything thereafter. We just have to select click run test, and it will run on its own. So while running, what basically it does is it gives different types of different set of stresses. Okay, display test stress. We can see these are the different stresses that they will be applying on the powdered sample. All right, and then they will be analyzing the flow behavior or the powder's behavior, and from that we will get a graph or the data. The data will look like, so I have already done it for you all. Flow function. This is the way the data will look. Okay, so in this product sample GIAN sample 1A. Sample 1A. So this is the test data, we will be getting, these are the stress points. These are, as I said, they will be applying at different stress points, okay. So they analyzed at around 35, I guess, yeah, 35 stress points, okay, at 35 different stress points, they analyzed the density. What is the density, okay. From that density we came to know, because this compression plate, what it will do is it will create a void, and then from that void it will create different stresses. All right. So in that stress, what happens is that they will see what properties or how this with the compression, how the powder is behaving. So in that manner, they will analyze the density changes, and thereafter, at last, we will be getting the gradient, the angle, cohesion, and values like consolidation, endpoint, major principal consolidation stress, unconfined failure stress, effective angle of friction, Hopper half angle. So these things, basically what they do is this helps us design the silos, or for the transportation, let us say, what are the the body or the angles that we have to maintain for the powder sample to stay stagnant or stay in a proper phase. All right. Now next we will be doing is, this process is entirely same. We will be using just this in place of the vane lid, okay. And here, of course, we will be changing this profile to the plain one. This is for the walls, okay. So here we will analyze what is the effect of the powder on the walls. All right. So the wall results, I will not be doing that again now. I will be showing the wall results only, the wall function results. So this is how the wall function results looks like. So they will indicate the maximum stress which they applied. Here also, like similarly, they will be applying, checking at different stresses. So here they use 31. So the difference, when normally the standard is 35, okay, they will run it for around 25 minutes. But if the machine signifies or finds out that there hasn't been any significant change, okay, after even after changing the stresses, there hasn't been any significant changes on the sample, so they will stop the test. So that's why here we saw only 31 runs, and there we saw 35 runs. Understood? So once, look, as you can see, the constant, the result is coming somewhat if you can see 640.7, 640, 639. So it is coming somewhere around 640 only. So it have stopped changing the densities. So they have stopped the test. Okay, then we will get the stress and densities, different significant stress and densities, and at last we will be getting the effective angle of wall friction and Hopper's half angle. Understood? From that end, we can design the material. We can decide what material we will be using, or what will be the angle of the walls that we will be needing for design purpose. I'm saying there are in various fields there are various applications as well.